So thank you so much for having me. So today I'm going to talk about biological patterns across domains and specifically interrogating information contained across these data sources. So I'm going to jump right in, but as I go, I'm going to survey the room and see how familiar you are with certain topics I'm going to talk about. And so that will give me a sense of how deep I'll go into some of the methods, and maybe I'll keep it more high level for certain topics. So. Multi-source data is everywhere, and I'm sure including on many of your laptops or PCs. In terms of publicly available data sets, this includes the Cancer Genome Atlas, where we have multiple different data types measured across many different tumors and patients. The Genotype Tissue Expression Project, which I'm sure many of you are familiar with, where they've measured SNP and gene expression data across many tissues. And the Cancer Therapeutics Response Portal. This is one of many databases of cancer drug response. So many different cell lines and measuring many different cancer drug responses in these cell lines. So leveraging multi-source data is really useful, for example, for understanding the stability of relationships across sources. So if you know that a relationship in one source of data actually is maintained across many different sources, it can empower you to really understand a lot more than what you've measured. So you can apply a specific model as a general principle. For example, a model for gene expression regulation in blood, maybe you can apply that to the brain if you know that that pattern persists across many different tissues. And this allows you to study difficult to access domains by proxy. And so domains can be difficult to access. For example, if it's, again, I'll use the example of brain tissue in people with psychiatric disorders, it's very difficult to access those cells. So if you can use buccal cells or blood cells, it would be really useful to know that you can use those to find relationships in the brain, for example. So two applications I'm going to talk about today are cross-tissue gene expression, as you may have guessed by my examples, and then cell line to patient cancer drug response. So the first one, I'm going to dig more deeply into the actual project and give you some teaser results, as well as set you up to hear more from my supervisor, Dr. Anna Goldenberg, next. And in the second one, I'm going to talk more about the theory underlining some work we've done there. So that one's going to be much more theoretical and diving into other people's work and how we will be applying that, as Anna will discuss. So first, cross-tissue gene expression regulation. Our motivation with this project was to leverage an existing data set, which is TCGA, to uncover patterns of gene expression regulation by regulator, tissue, and gene. So as I said, we used the Cancer Genome Atlas, and the reason we did this is because there's multiple different regulators that we can measure in this data, and they're measured in the same individuals. So we can see relatively what role they play in gene expression regulation. And we did this by creating models for all of these regulators and relating them to gene expression across different tissues. So we wanted to find the most relevant per gene regulator, and so this is what I'll be talking about. So that's, say, microRNA. How important is microRNA for the gene expression regulation, or methylation for gene expression regulation, for example? And then Anna will cover some regulatory effects across tissues. So can we compare the effects of a single CpG, for example, across different tissues? And then last, implications of regulatory hits. So if we build these models using the same CpGs in multiple different genes, can we kind of turn it on its head and say, now we have a specific CpG site, how important is that if we want to link it to genes? Which genes are the most regulated or associated with this CpG site? So we created a resource to do this called IMODIS, so Integrative Modes of Estimated Gene Expression Regulation. And we model regulation on a per gene, per tissue basis. So we're independently modeling each gene in each tissue across over 16,000 genes in 21 different cancers. So to go more deeply into the data that we used, I already listed the regulators that we're considering here. But here are the 21 cancers that we actually use. We combine renal adenocarcinoma and colon adenocarcinoma, as has been done in previous TCGA exercises. And we use an array of different technologies that have actually measured these regulators. So in terms of quality control, we removed low quality samples, of course, samples without all data types so that we're fitting each model with the exact same individuals, and then related individuals and non-Caucasian individuals. So this is something I kind of want to take an aside to talk about. People often forget that there is healthy germline SNP data in all of these participants in TCGA. It's how they got the somatic calls for the CNVs. And so we actually use that germline data to do PCA and to find cryptic relatedness in the TCGA data set. And so this allowed us to actually create a more homogeneous sample. But you can see that in some of these data, in some of these cancers, you're seeing maybe a smaller sample size than you would expect. And it's in large part to this QC step where we're removing individuals. So particularly BRCA, that was a lot of non-Caucasian and related individuals that we ended up removing in that step.
So is it more ancestry or
it was
close relatedness that you're dealing with?
It's ancestry and close relatedness. So the ethnic stratification was definitely took a toll on the sample size, but I actually found more relatedness in that sample than I expected. So a bit. Yes.
Keep one individual from
yes. Yes. And actually, interestingly, for the ethnic stratification, we used PCR. So it was a really interesting. I've just used PCA, like a standard PCA, to do that ethnic stratification. But this technique actually finds a set of mutually unrelated individuals and estimates your PCs based on those. And then because I kept in cousins, for example, in the sample, I was able to then have a better ethnic stratification PCA and remove non-homogeneous individuals from it using that technique. And then all the data was transformed to a standard normal. So this is different for each of the data types. They follow a different distribution. So that step looked different in many of the different cases. And then the data was corrected for age, sex, batch, histological subtype, tumor nuclei percentages, as well as 2 SNP principal components. And these PCs were really important for us as well. So those are what we fit using PCR. And so you can see here, though, we would have removed a lot of the variation in our data. And this was intentional. We wanted to estimate how much variation can we explain on top of these known and measured features of the data. So even though this removes a lot of variation, that is something we really wanted to do here. Yeah?
How did you correct for those?
Just a linear regression, and we did it in the regulator and in the gene expression outcomes, so it's akin to just keeping it as a covariate in a linear model.
With fixed or random?
Fixed, yeah. All right, and so then we did a biologically informed feature selection. So obviously, with this number of samples, we couldn't use all of methylation probes or all of the SNP sites to create our models of gene expression with as few as 100 samples in some cases. So we had to select down to a subset of these samples. And so the way we did this was, in the microRNA and TF expression case, we selected them based on prior evidence. So TargetScan is a database that, based on 6, 7, and 8 mer matching with the 3' UTR of the gene, they've linked microRNA transcripts to genes. So we use that as like a targeting matrix, and then only included microRNA in the model that are said to target those genes for each gene's model. And then ENCODE has a similar matrix that they put out where they have evidence that the transcription factor, a given transcription factor targets a given gene. And so this is very cancer focused, but because we're using TCGA, we thought that that was okay. And then for lincRNA methylation and SNPs, we looked at the 1 megabase around the gene body region, so upstream and downstream from it, and included all of those, anything that lies within that. And then for copy number variation, it's the mean copy number variation of the gene, so that's 1 value. And then with each of these, we fit an elastic net model with every combination of the regulators. And so that's 64 models per gene per cancer. And then we evaluate these models using a held out sample predictive accuracy. So that is the press R squared. It's a R squared value, except the error sum of squares is a sum of squares based off of a held out sample. So it's the prediction in a new sample where the model hasn't been used with that sample, or the model has not been fit with that sample. And then this allows us to summarize the effects by tissue, gene, and regulator. So now let's look at what this exercise actually produced in terms of results. So I'm only going to talk about, again, these most relevant per gene regulators, but Anna will go more in depth about other results we were able to get from this analysis. So first, just to orient you here, what we have on the y-axis is the added predictive accuracy. So we're looking at every combination of regulators as a background model. And let's start with microRNA, for example. Every combination of regulators that are not microRNA, and we say, when microRNA is added to that model, what's the gain in predictive accuracy that you get?
So this is averaged over the 32 models.
Yes, so this is a median. So this is the median, but in effect, yes, it's a summary of those. And so each point here is one gene's median gain from that regulator being added to all the other 32 models.
Taken over each of the other ones being turned on.
Exactly. So in the case of this specific gene, lincRNA explained all of the variation, but as you can see, also that's a very rare case. These box plots assume a standard normal distribution. So the outliers, these points here outside the box plot, show that it is not normal, the data that we get out of this, and in some cases seems almost uniform across the spectrum here. So one thing that immediately pops out is transcription factor co-expression is very predictive. And this is something that we would really expect to see. This is why you control for peer factors in your models. When you're doing EQTL analyses, this is effectively representing the cell state. So this was nice to see just as a sanity check that this is very predictive. In the case of methylation, actually, we see something really interesting. In a subset of cancers, we have a larger gain in predictive accuracy than we have with transcription factor co-expression. And just zooming in on this a bit more. The particular cancers that this is happening in are breast, cervical, esophageal, and head and neck squamous carcinoma. So that's these first ones here. Then this is lung squamous carcinoma, and this is sarcoma. And almost all of these cancers have a squamous histological subtype within them. And previous work in the TCGA data has found when you're clustering across multiple modes of data, so microRNA, methylation, gene expression, et cetera, they found that these cancers do tend to cluster together separately from the rest. So it seems like we're detecting a similar signal here. And the other nice thing, yes?
What does negative mean?
Yeah, so negative means, actually, I'm going to jump to the next. So SNPs is a really nice case of like negative gain in predictive accuracy. So negative means that because this is a penalized linear regression, it means that you have so much noise added when you add that data type to the model that the penalty term goes very high and it squishes all of the coefficients to a lower value. So even if you had something in there that was very predictive, the noise you've now added by adding that data type is just making it so that you lose a lot of your predictive accuracy. Yeah, thank you for asking. Yeah.
What if for your SNPs, correct, you just, the only constraint you had is minus plus
Yep.
1 megapixel.
Exactly, yeah.
But what if you use annotated chromosome regions and then only keep the SNPs that fall within node enhancers or enhancers cross promoters?
Absolutely. So we played with a lot of those ideas, and at the end of the day, we were just worried about biasing ourselves too much by doing that. So we think that would be a really interesting next step here. But we wanted to see if we just agnostically say this whole region, throw it all in and see what happens. But I think it would be really interesting to see now if we subset it, maybe we're not losing as much.
Yeah. So basically, where I'm coming from is the fact that you are saying TFs have a significant effect.
Mm-hmm.
Those TFs are going to be binary promoter regions or enhancer regions.
Yeah.
So if you restrict your SNPs to the ones that fall within those regions, you may actually gain accuracy.
Absolutely, I wouldn't be surprised. The other thing here with the SNPs is in cancer, a lot of other data types are explaining a lot. So when SNPs are added to the model, there's a lot to lose. in a sense. So that could also explain this drop here, that there's a lot of other data types that have a lot of power in predictive accuracy.
And one last question.
Yeah.
So is there any, let's say, preliminary step to predict causal SNPs per, let's say, region? So let's say you have multiple synonyms. So do you predict which one are the likely causals from your SNPs?
We don't in this case, but I think that would be really interesting to look at, yeah. And sorry, I think there was a question in the back, or maybe that was us? No? Okay.
In your regularized linear model, do you have the same like lambda penalization parameter for each class when you put them in together?
No. So we tried that, and it just obliterated our predictive accuracy. So yeah, so these are all, the lambdas are fit in a 5-fold cross-validation step.
And then one for each class?
For every single model that's fit.
For every single model, each group gets its own?
Yeah.
Okay. So it's weird, do you think you lose a lot for SNPs because there's so many SNPs?
Yes.
Okay. That's
Basically, and that's why I think the suggestion here of subsetting it to certain regions would actually lead to a large boost in that predictive accuracy, because you don't have all the noise kind of in between that's being added.
What if you use something like Cosmic to force it down even smaller?
Yeah, absolutely. So this is, as like a first step, we wanted to just be as inclusive as possible, and then as we add more filters onto these data types that we include, then we can have this as kind of a reference that we're starting from, and then see how much we gain by that subset, or maybe lose even. And so on the topic of SNPs though, because we also have normal sample data, so in some cases it's blood, in some cases it's tumor adjacent normal, we can take these cancer models that we built and apply them to the normal tissue. So that's what we did here, and we see that the SNPs actually aren't losing as much here. And I think this is goes to the fact that our methylation and microRNA gains are much lower in normal tissue, which I think is to be expected based on cancer biology, but we see that TFs, the TF co-expression is still quite high, which again, if it's reflecting the cell state, this is to be expected. All right, so now to jump to a specific gene, so we can zoom in on specific genes in that full graph. And here, to reorient you, we're still looking at added predictive accuracy, but here each point, instead of being a gene, it's a specific model, which is really nice because when we see these long stretched out box plots, it actually means that in some cases, you see a low gain in predictive accuracy, and with other background models, when you add that regulator, you see a large gain. So now we can start to tease apart shared variation that's being explained by different regulators. So CRIB2, crystalline beta 2, it's expressed in tissues across the body, implicated in behavioral phenotypes as well as cataracts. But what was really cool here is we see that SNPs are adding a lot, and seemingly in most cancers, so 15 cancers, we see a median increase of between 25 to 75% variance explained when adding these cis SNPs to the model. And we also see that in those models, it doesn't really matter what the background is. Because those box plots are so tight, it didn't matter what was already in the model. And that makes sense when you look across the other regulators, it's pretty flat, except in this special methylation case. So here, it seems that SNPs and methylation are actually explaining some of the same variation. And we can actually test this. So this is breast and esophageal cancers. We can test this by now only looking at models where we already have methylation added. So now what's the gain when you only look at models that have methylation included? And we see that conditioning on the methylation effectively removes the predictive gain that you get from the SNPs. So whatever the SNPs were explaining is also being explained by methylation here. So some limitations and considerations of this is, yeah, the results are as good as the data going in. So it's really in the context of the data that we've chosen. So as you brought up, how we select that data is really driving our findings. And this is why we wanted it to be really agnostic and not putting any priors on it. But now I think is the time to actually put those priors on and investigate if we can get a boost from doing that. There's many choices along the way. So we played with a lot of ways to model this data and ended up with a really intensive data mining approach. And And we considered doing a kind of random effects variance partitioning, but we wanted to get the covariance between the different data types. So we moved away from that and thought this was an easier way to tease apart that shared explained variance. And then last, many findings are difficult to directly validate without comparable data. So some of the signals we find, so for example, CRIB2, it's come up in a lot of EQTL analyses, and it makes sense based on our findings that that would be the case. But without this multimodal data, it's hard to actually know that this is exactly replicating in a new data set. So we do our best with running many experiments in parallel and seeing where are our results, where's there a consensus across tissues maybe among our results, but it's still a difficulty here. All right, so now moving on to something pretty different from the last bit, is looking at domain adaptation in cell line to patient cancer drug response. And at this point, I just want to kind of survey the room and know how many of you are familiar with unsupervised domain adaptation and the literature around that. Awesome. So that's what this next section is going to cover. So those of you who are really familiar with it, jump in at any time if you have comments about it, and then feel free to ask questions for anyone who's not familiar with it. So the motivation here is that, and this is going to the measuring something by proxy, cell line drug response is pretty easy to do. It can be done in massively parallel experiments, and it's relatively cheap compared to patient drug response, where you can't actually replicate across patients, of course, and that patients' death or their response is very costly. So it would be great if we could find a signal in the cell line that can be transferred to the patient. So a solution that Anna's going to talk about in her talk was to use unsupervised domain adaptation. And so to give a background to that theory and some algorithms that have been used to solve this problem, I'm going to talk about unsupervised domain adaptation more broadly now. So unsupervised domain adaptation has the aim of training a classifier using a source domain. So here I have the source domain where you observe your x's. So you can imagine this is x1, x2 axis where you've plotted your x's. And you also observe your labels in this source domain. So the colors here represent the labels. But you really want to apply your model in a target domain. And in this target domain, you're observing the x's, so this xy axis is observed, but you're not observing the labels there. So the colors there are put there for our benefit, but these would be unobserved in an unsupervised domain adaptation case. And the approach you really want to take is you want to minimize your source error because this is the only place where you actually have labels, so you're going to need to minimize something. But ideally, you want to minimize it in a way that minimizes your target error as well. And so here you can think about the source as the cell line drug response, and the target as being the patient drug response. So what has been done theoretically with this work is that instead of minimizing the source and target error, you minimize the source and an upper bound on the target error. So now I'm going to talk more about that upper bound and the development of it and what it actually means for how algorithmic solutions have solved this problem. So I'm going to talk about Shai-Ben-David's lower or upper bound on the target error, but this has been work across 2007 to 2010 by Mansour et al., Ben-David et al., and many groups really around this time were developing this same bound and kind of converging on the same thing. So first, this first term is the error of the target error under a given hypothesis. And the hypothesis here is a given classifier. So you want to minimize this. And the bound on it is a sum of your source error under that same classifier, the h distance, and that h distance is actually a function of the minimum domain discriminatory classifier effect. So that is to say, your ability to distinguish between your source and your target domain. So this is a classifier that distinguishes source and target and not your labels. So it's not your hypothesis, it's effectively a distance between your source and target domain. a complexity term. So this term encapsulates the probability of the bound, your sample size, and your VC dimension, so your complexity. And then this last lambda term, this one's very important. And as I'm talking about solutions and problems with some of these solutions, this is something you want to keep in the front of your mind. So this is a theoretical term that's the error of the ideal hypothesis across both your source and target. So if there is no line that can be drawn that perfectly separates your data, you're bound by that issue. So your bound is as good as your best classifier you could possibly draw, encapsulating to 2. So some solutions that have sought to minimize this target error, either directly or indirectly in their implementation, are importance weighting across source samples, mapping the source to the target domain, and then finding a common embedding for the source and target domain. And in every case, they're really looking at this H distance. But the H distance is fixed. Your data is what it is. The distance between them is, between your source and target domains is what it is. So the way that these deal with it is, in the first 2 cases, it's actually as if you put a function on the source domain in order to minimize that distance between the source and the target domain. And in the case of the third approach, where you find a common embedding, you find a function that will combined make the source and target domain both closer. Yes?
Is this like a distance between distributions?
Yeah, yes. So first to talk about importance weighting. So here it's work by Shimodera et al. In 2000 that has been used and changed by many others. So in every case of these examples, I'm actually just going to talk about 1 or 2 papers, but know that there's a vast body of work around all of these. So here, instead of optimizing this loss term, the standard loss term, what Shimodera et al. Propose is optimizing a weighted loss. And the weight is actually based on the overlapping support between the source and target domain. So it's saying, actually, if you build your boundary where your source is overlapping your target, you're going to find a better boundary, is their contention here. So obviously, when you don't have overlapping support, though, all of these weights go to 0, and you can't do this. Another approach is mapping source to target, and I'm going to talk about it in the context of cycloGANs and cicada, just because I think this work is really fascinating in terms of generative adversarial neural networks. So a cycloGAN is actually, I'm just going to pull the room. How many people are familiar with generative adversarial neural networks? Oh, awesome. Lots of people. So I'm going to go over it very briefly. So work by Goodfellow et al. Introduced this idea that you have a generative model where you produce some fake data, and you have a discriminator that is discriminating between fake and real samples, and that's added to your loss function that you pass back. So here, what cycloGAN does is it does that, except it's a conditional GAN. So your input is going to be, for example, your input could be this summer image, and you say, I want a winter image, so generate a new winter image. So cycloGANs, it has the adversary there saying whether this is a fake or a real winter image, but then it goes one step further. It now takes that generated winter image and generates a new summer image, so cycles it back to the original, and then sees how close that original is to your input. And so that's the cycle consistency loss that they actually add in there. And it's led to some really amazing results in terms of a conditional GAN performance. But Hoffman et al. Actually integrated this into a unsupervised domain adaptation problem. So here what they do, this is the image from the paper, but in effect, they train a cycloGAN, and then they use that cycloGAN with the label on your target or your source data to now just train the classifier in the new domain, so in the target domain. So if your labels are consistent across the 2 domains, then this works. Obviously, this will be constrained by that fact. And then the last one is finding that common embedding for the source and target. So here I'm showing the deep adaptive network from Long et al., where they place their features from a neural network from the source and target data. So they pass both source and target data through these first few layers of a neural network and then embed it in a reproducing kernel Hilbert space. And then they find the distance between the features in that space. And so in effect, they just create a penalty term on the distance between the features that are output between the source and the target domain. And so in so doing, mapping them to a common domain where you build your classifier. This can be done another way as well, though. So this is domain adversarial neural networks by Gannon et al. And they also developed the gradient reversal layer here. So here, instead of having a distance metric between your two domains, you have an adversary that is distinguishing between the two domains. So it's similar to the GAN approach, where you have an adversary discriminating between real and fake, except here, the adversary is discriminating against can I distinguish between the source and target domain based on the features that your model has output? And then, with those features, building a classifier. So some limitations. So this has been a really fun year for limitations in unsupervised domain adaptation. In quick succession in 2019, 3 different papers at least have come out talking about the shortcomings and constraints to unsupervised domain adaptation. So I'm going to briefly talk about them and talk about some real-world scenarios when you can imagine these happening. So all of these methods are really relying on an identical marginal label distribution, something which is not talked about a lot, covariate shift alone, which is discussed much more often, and overlapping support, which for which there's a hilarious example given in the paper that I'm looking forward to going through. So first I'll start with the identical marginal label distribution. So Wu et al. This year pointed out that if you have different marginal distributions, as little as that, between your source and your target domain, you're going to be misaligning your data because you don't have the benefit of the labels, so you don't know that you're misaligning it as well. So here, going back to the example I showed, now let's say there's a few more greens than there are blues. Now if we align it, we can see that the greens are now encroaching over into the blue space. So automatically, if you build a classifier here, you're definitely going to be misclassifying a share of your target domain, guaranteed, if you're aligning them with that domain adversary, if you're doing it with a constraint on the feature differences specifically. The next one is covariate shift alone. So if there's a shift in the conditional relationship between your source and target domain, then you're going to run into big issues. So I'm showing a very dramatic example here. Let's say the greens and the blues are swapped, and now we find a latent space that we map these both to. Obviously, we find a perfectly wrong classifier for the target domain based on our source domain. So this is a really extreme example. One case in which you might see this is if you have an assay where one group is the base case and one is one's a control and one's a case in one group, and then you swap those labels somehow in your other group. Because cases and controls are not always so clear-cut in biology. Another situation where you might see this is actually more subtle. Maybe it's a change in how you actually define your outcomes between domains. And so this is something that Anna's going to talk about a bit more in terms of cell lines versus patient drug response. It's a slightly different outcome that's being measured. And so when you're aligning those domains, you might actually not be aligning them quite appropriately. And then the last one is overlapping support. So I really love this. Johansson et al. Have this figure in their paper, and I think it does a really nice job of showing how this can be a problem. But I'm going to recapitulate it here with the example I've been using throughout the talk. So here I've swapped the locations of all my source and target domains. So let's say the source is lying in those quadrants of the graph, and then the target is lying here, and remember, we don't have the labels for the target. So there's 2 easy ways to align this data. The first way is perfect. So great, awesome, we aligned it, our marginal labels distribution is matching, and we get a really nice classifier. But we could also align it in the exact wrong formation. So we get a perfectly imperfect classifier. And without labeled samples in the target domain, we can't know what we're doing. So when there's not overlapping support, we're really prone to this kind of error. So Johansson et al. Actually don't propose a solution to this. They more just propose a bound that includes the overlapping support within it. But this is a real problem and something to really think about, especially as you're working with very high dimensional data. The likelihood that you don't have overlapping support may be quite high. So, yes?
Do you think with few-shot learning would solve these type of problems? Like, not instead of completely unsupervised, maybe you have a couple of labels in the domain, and then can you resolve this problem?
Yeah, I don't think it will completely resolve it because you're still holding the assumption that with those few labels that the rest of the data in, say, the target domain is still following that distribution. But I think any labels you can get, this really motivates, like any labels you can get in that target distribution are gonna help you with this alignment because this perfectly imperfect alignment, it would be way less likely to happen if you had those labels. So absolutely. So some takeaways, I would say the Ben David 2010 bound, it should be handled with care. Many algorithmic solutions are focusing on that h distance to the neglect of that lambda parameter, where if you now embed it in a space where it's not even possible to, like your perfect classifier that you could get is giving you maximal error, then you haven't actually done a good job of domain adaptation. So I think my own recommendations are use it when your shifts are relatively small, so favoring overlapping support, or where predictive validity can be closely monitored or isn't as much of a problem if you make errors, or you can test those errors very easily. And then when you're unsure of these things, yeah, exactly as you said, get target labels. Assess the stability of your classifier, and really characterizing your domain shift, I think is very important here. So really understanding the shift and the assumptions you're making when you're modeling that shift. Sorry, yeah?
So how do you characterize the domain shift?
Mm-hmm. So, yeah, good question. I think plotting your data, I know that sounds so ridiculous, but I know a lot of people don't do that. Even actually seeing how, if you can classify between the two groups, so finding the actual h distance between the two groups is a good way to do it. So yeah, just looking at your data and testing your data for differences between the domains. All right, so I just want to say thank you so much for the organizers, for having me, for my supervisor, for bringing me along when asked. And then, of course, many people in my lab here and our funders. And if you have any further questions, I mean, of course, ask me now, but my email is there as well. And yeah, thank you. So, is it
Questions?
questions? Any?
Looks like practice being failed. I have one more.
Yes.
Can you explain why it's called overlapping support? I don't think I quite, I mean, the example was really clear, but what is the condition that you would like to be satisfied, and what is it to do with this?
Interesting.
Or is that, maybe the question doesn't quite make sense, but like, why is it called overlapping support?
So I am using Johansson et al.'s term for it there, and it really is like support in terms of a statistical distribution that your probability distributions overlap given the data you have.
Like the distribution in the source and the distribution in
Yes,
the target?
yes, yeah, of your x's. That makes sense. Okay, I hope that was. Yes?
Really good talk. I'm wondering, so in your domain adaptation case,
Yeah.
if there's any way of telling how failure to adapt to a certain domain will tell you anything about the specific biological representation?
Yeah. So I don't think it can tell you what's going wrong, but you can know by the fact that something is going wrong that you should search for that thing. So I think Honest Talk is going to be a bit of a journey on that. I know when we applied it in certain cases, we found that we were very underwhelmed by the results, and we've talked to others who have been as well, which really made these papers coming out this year kind of a blessing, like a sanity check in a way. And so I think when that happens, you really want to start thinking about the assumptions you've made in your data and exactly what shifts may be existing that you maybe haven't thought about before. But I don't think it can tell you what the problem is. I think it's quite situation dependent.
So
Yeah.
we talked about marginal difference, marginal distribution difference
Yeah.
in this effect. What if it's even more extreme than that? That is to say, one data has one additional subclass, right?
Yeah.
Label, where the other one doesn't. So it's technically marginal distribution difference. One is 0, the
Yep.
other isn't. But are there any work that talks about domain adaptation when your domains have inherently different, like basically subpopulations?
Yeah, thank you for asking. That's actually a really interesting body of work of like open set domain adaptation. So saying I've trained my model with these labels and now I want to apply it in a case where I know there's more labels. And so in effect, how that's been done, so Sato et al. I believe has some work in this, or it might be Seiko. And then there's a universal domain adaptation paper, I think it was from this year or 2018, that does a really nice job on that. And effectively what they do is they threshold your certainty of putting it in either class. And if you're low certainty in both, then then it's a new class and they create basically a new label in your target domain with that. But yeah, it's a very interesting body of work, and there are many scenarios where you can imagine that's the case, so very useful. Yeah.
So how do you tell the difference between cases where there's just not a big separation between classes versus the fact that you might just need more data? I mean, assuming that you're powered statistically.
Yeah, I think that's a really situation-dependent theoretical question. So if you're looking at two domains and you're like, these are really just different groups, that's a different thing than I have a small number of people measured in this group, and it could be that as I measure more, it will have overlapping support. I think it's hard to answer that in general, but I can imagine in many scenarios, you may just want to get more data to see, unfortunately. Yeah.
Thank you.
Right, thank you. Yes?
Your experience, you show different approaches, right, to deal with domain adaptation. But in your experience, which one worked better, or is there one that worked better?
Yeah.
Or your particular domain?
So I would say that they all do a pretty good job if the assumptions are satisfied. I think that mapping them to a common embedding, which is what Anna's, like what she's going to talk about in the work of my colleague Ladislav. So mapping it to a common domain is a totally reasonable approach. I've heard that people who've worked with the gradient reversal layer, they actually have better luck if they don't do it. So gradient reversal layer passes back the error in tandem with your error on your classifier. And I've heard actually alternating in the style of kind of a GAN classifier is a bit more effective here. But I assume there's some nuance to it. But yeah, actually, like even cyclogans, I think they all should work well. I think that it depends on a case-by-case basis, unfortunately. But they do a good job if the assumptions are met. Yeah. Great, thank you.
So thank you very much for inviting me, both seminars. It's very exciting to be here. And thank you all for coming. This was a lot of fun for Lauren and I to work on and to give you some ideas. Just to give you a little bit of background on where I come from. So I work in both spaces, in computational biology, but also in computational medicine and healthcare. I'm currently leading, co-chairing an AI in medicine initiative at SickKids for translating AI to the bedside. So this is one of my passions now, is how do we get methods and approaches that we can adopt, that the clinicians will benefit from and ultimately the patients will benefit from. So while I'm not talking about a lot of that work today, I will mention some of the projects that we have that are in that space. If you are interested more in those, I'll be happy to chat with you after. I'm also an Associate Research Director, Health at the newly established Vector Institute, which is an AI, one of the 3 AI institutes in Canada. So that said, as I know quite a few of you were in Lauren's talk, there's a tiny bit of an overlap on the key points, but I will try to give you kind of a more summarized view of the several projects that are in the domain of building predictive models for explaining biology using machine learning and trying to do so responsibly. So it turned out we as computer scientists, so I have to tell you that I'm a field fundamentalist in machine learning and a computer scientist, and this is the place where I come from. Having been in this area for the last, 12 years, I'm starting to feel like I can weigh in biology, but certainly this is, I'll be happy to hear of any feedback. But there are still things that we are discovering that are surprising. So it turned out that we know a lot about just a few genes, right? We know that there are tens of thousands of genes, but these 10 genes, so this is 10 genes from Dolgin's paper in 2017, these are referred to in literature and kind of explored in 45,000 papers. So obviously an estimate, but TP53 in 10,000 papers, right? This gives you a sense of, I didn't put the end tail of this distribution, but there are a lot of genes that are never mentioned or mentioned once. So the regulation of these genes is not known. It's not known how they contribute to the biology of the cell. So this is one of the reasons for the project that Lauren has spearheaded that I will talk about now. So when I started in this field in biology, I quickly absorbed this nice common beliefs, right? The methylation of the promoter affects the repression of the gene. MicroRNAs downregulate gene expression by binding to the 3' UTR. CNVs can increase the activity by having more copies of the gene, et cetera, et cetera. And as I was digging more into it, I would find counterexamples to this. And I would go to my biology friends, and they would say, yes, we knew that. I'm like, but why are you selling these stories? So this is one of our attempts to kind of shed a broad light onto the picture. So we already know for sure that even the budding computer scientists in biology know for sure that EQTLs across tissues are different. So for the same genes, they may have different EQTLs. They may have different enhancer regions. There are tissue-specific gene expression patterns and gene expression regulation. We know that, right? What we don't necessarily have a handle on, and I don't believe this is known broadly, is how big is that heterogeneity genome-wide, right? What is out there? So this is just a teaser. For those of you who were at Lauren's talk, you will recognize some of the plot, but this is just a teaser. So these are 2 genes. One is very well known, EGFR was in the top 10. For those of you who do breast cancer, you know it very well. And LEFT1. LEFT1 is a ligand in TGF-beta pathway, one of TGF-beta ligands. So you can see here, this is the regulation of these 2 genes by microRNAs. And you can immediately see that the patterns are very, very different. In EGFR, the pattern is complex, whereas in LEFT1, it's actually not, it basically doesn't look like microRNAs across tissues is actually acting much or contributing much to explaining gene expression. But in one of the tissues, in sarcoma, it looks like it's contributing a lot to explaining, almost 100%, right? So in one of the tissues, there is a very strong understanding. And I can tell you, we picked this gene for its name. It kind of looked cute. But this is just there's so many genes that some of you are potentially studying this gene, but like these are not broadly known facts, right? So going further with the LefT1 and EGFR comparison, it turns out that LefT1 is primarily regulated by link RNAs, right? Whereas EGFR, not so much. So you can see that there is a large variance around the models. So in some of the tissues and some of the models, it definitely contributed more than others. But it is a very striking difference in how these two different types of regulators are contributing to the regulation of these genes. And finally, the CNVs, right? This is kind of, EGFR is certainly, CNVs and EGFR genes are a known biomarker. In LefT1, the CNVs are quite silent. So this gives us a sense of just how different this different regulator types are weighing into the regulation of the genes. So how do we learn this about thousands of genes across the genome? Fortunately, there is data. And one of the things that I can tell you as a machine learner and as a person who builds a lot of predictive models, what we usually do is we take the patients and we, as our sample size and all the genes as our features, and then we try to predict something. And there's always n is much greater than p, and we're always underpowered. It's always very frustrating and multiple modes, and we can't really find the same gene. So the idea here was to say, we will do it per gene. We'll do it per gene. We will build a model per gene. And in this case, we have a lot more data and a lot fewer features, and then we can actually do something about it. So we chose TCGA for the obvious reason that, to my knowledge, and please correct me if I'm wrong, I would love to know that, but to my knowledge, this is the only repository that has consistently measured all of these different regulator types for the same individual, for the same sample. So you can see here, this is the human that you already saw before. This is the colors represent the different tissues and the different cancers. And we basically use the data directly from TCGA. You will notice that the sample size for each of the tissues is much smaller than what you would see if you just log into the portal. And this is due to a very rigorous QC that we did. And Lauren has spoken much to that QC, so I will not cover it here, but the QC here is, of course, very, very important. We have to really trust that we are not recovering some population-specific or sample-specific relations here. We want to have this broad information. So we call this in a modest resource. It's an integrative modes of estimated gene expression regulation. It kind of came after, the explanation came after the name. But we did build a model regulation per gene, per tissue in over 16,000 genes in 21 cancers. So what can we learn? We built 64 models per gene, where we look at all combinations of all the regulators. So starting with a single regulator and looking at all the combinations. So what this allows us to see is not just the marginal effect of each of the regulators, but also the contribution to the joint and how the regulators interact. So I've put up TP53. My students and colleagues told me it is dangerous because there will be people in the audience who know the regulation of TP53 super well and they will not be happy with you. I think that we can still learn even about the regulation of TP53. So this is one of the things that we can see here. So for example, this, the first one is a microRNA regulation. The fourth one is the methylation regulation. So you can see that the microRNAs by themselves do not seem to have much of the explanatory power towards predicting gene expression. So I didn't mention, sorry, this was in Lauren's talk, that each of these models is predicting gene expression. And what we are representing here is this predictive accuracy of each model.
So is this across all cancers?
So this is across one tissue. This is one tissue. an example. This is the predictive accuracy. This is a R squared on a held out sample. So you can see, this is already interesting, right? That by themselves, microRNAs and methylation don't explain as much as them jointly. So you know that there is some kind of a joint effect here. We notice that when we add C and V, so again, C and V's by themselves are not contributing much to explaining the expression change in the variation and expression in TP53. Jointly, they explain more. So there is effect that C and V is explaining that is beyond methylation and microRNA effect. Notice that we would expect transcription factors to have a larger effect. And in reality, they have as much an effect, a marginal effect as methylation. But when you add transcription factors in addition to the methylation when it's already in a model, transcription factors don't explain as much. So this is not about the expression of the transcription factors. This is about that region, the proximal region, the cis region being methylated, which I think is very interesting. Yes, there was one over there, then here.
Can you talk about the type of the models that you use for predicting the gene expression?
We use logistic regression. So we use an elastic net, yes.
Okay. And then regarding this additive effect of the methylation with the TF, does it mean that, transfer factors are sensitive to DNA methylation or for gene methylation or?
Sorry, can you
okay.
describe?
So I missed the part that, so you have the DNA methylation, and also you add the TFs, right? And then you gain a power in explaining the gene expression. So I was wondering that is there any underlying biological processes that you are captured by joint modeling?
So that's our implication, that there is an underlying joint process. There is no directionality in this model, so we can't say that it is methylation came first and transcription factors came second or anything like that in the model from our modeling. But we do know that there is there is no explanatory power in transcription factors beyond having microRNAs, CNVs, and methylation in the model. So I think that's something to help design the next experiment to figure out what is the actual underlying biology. But it helps you to focus on the specific types of the data to collect. Yeah, I think the second one was here.
How many microRNAs do you consider here? There are over like 2,000 microRNAs.
So this was mapped by 6, 7, and 8 microRNAs from target scan per gene.
No, like target scan finds the target genes, but how many microRNAs?
We used all the microRNAs that were mapped to the gene based on the 6, 7, and 8 mers.
And you use the expression of the microRNAs in conjunction with the fact that they target that gene.
Yeah, that's the model. So all the microRNAs that were mapped, we use the microRNA expression from the tissue, from the cancer to predict the gene expression.
So the similar question, so for the transcription factor, also the gene expression level of transcription factor, rather than transcription factor binding sense, right?
Gene expression.
Okay.
Yeah.
There are fancier models where you could use the predicted activity, the protein level activity of the TF rather than the RNA expression of the TF. Do you use
predicted activity?
Yeah, you could basically, through the targets, infer the protein level activity of the TF, which is not always the same as the level of mRNA expression of the TF. You don't do that.
We don't do that. We use the accessibility by the encode to figure out which TFs to use, but we don't use any predictive. So basically, I know what my models do. I don't know what other predictive models do and how good they are and whether they've been validated with respect to this particular setting and this particular tissues, and I don't know how good the accuracy is. So for me, it's hard to then attribute, right, where the variation comes from, whether it's due to the error in a predictive model or so. That's why we use it as a proxy.
The RNA level expression of the TF is indicative of the activity of the TF.
Yes.
That's okay.
It's a proxy.
Yeah, that's good.
Yeah. Last question.
Yeah. Do you have a sense of how correlated these different factors are in your particular data set?
They are definitely correlated. So we are using ElasticNet as a model for predicting gene expression. So it's okay to have correlated features, but this is, thank you for leading me to the next slide. We do know that expression in TFs will be correlated with the expression of the genes, and this is what's shown here. So when we average all the models across the over 10,000 genes for which we had all the same data, you actually can see that TFs come out much more so as predictive and explanatory of the gene expression than any other regulatory type. Yes, so this was the last question for that slide, but please keep asking questions, no problem. As you can see, Lauren mentioned, but I see that there are some questions in the audience from people who weren't there. So this is an average. So this is basically a slide that recapitulates some of the stories that we know, right? And for example, we can look at the methylation, and we can see that there's quite a bit of variation in the methylation and across tissues. And what we found here, for example, that the squamous tissues actually have a lot more methylation implicated than other tissues. And this makes sense because squamous carcinomas are associated with environmental effects a lot more than adenocarcinomas, for example. So this makes sense, but there is a variation. I think what's interesting here is that microRNAs have a huge variation in how they affect genes across tissues. So every single, almost every single tissue has a full range of models that go from completely predictive to not predictive at all. And I think this is very important to know, that when we are talking about the weight of the microRNAs, it really is gene-specific, and we have to be aware of that when we are contributing this information.
Just to make sure I understand, the variation you're showing here is not per microRNA, it's per for each different model that incorporates, it's per gene.
This is, so each point here is a gene,
Okay, so
and
for
this
some
is
genes, microRNAs are very important. For other genes, microRNAs are not important.
Right.
Got it.
Each one is a gene, and it's the median over the other models.
Yes. Thank you. So Alex was at Lauren's talk, and he listened well. Yes.
So one question. We talked about restricting SNPs, but one additional idea is, could it be that relationship between SNPs and genes are highly nonlinear as opposed to the other ones, because a lot of them are mediated through enhancers or additional regulators, which may fan out the effect in a nonlinear fashion. So are there any work that you applied about those elastic non-linear models? Let's say any type of non-linear models that you try to see how.
So I have multiple answers to this question. One is that the linear models are quite popular. So PredictScan and everything else uses linear models. I think that having non-linear models might not necessarily yield more information into the actual effect on the expression. There are epistatic models out there, and we've done some of those, and it's just, they're difficult to interpret, and they're subject to the assumptions made by the models. So I think there is a reason. I feel like as a machine learner, I get excited about complex models, but as a scientist, I feel that sometimes having the simplest assumptions gives me the most insight into my findings. And I think the goal of this particular work was to try to get the most insight. So having the bias of making a decision on the linearity, we can actually make certain assumptions. Another thing about these models is that we are actually looking for a lot of direct effect and direct understanding, like you saw in the example with T53. So if SNPs are acting through methylation or through other factors or through a binding site, for us, it's less of a direct evidence. So that is to be explored next, right? if there is a, SNPs that are implicated in there are genes, for example, the CRIB2 example that Lauren gave, that are really strongly weighing into the regulation of gene expression, right? So there are genes that certainly have the effect. This basically tells you that SNPs a lot of times act as noise because there are so many, because they are hard to sift through when there is other stronger signal, but it gives you a sense of where to go next with the experiments to run. Yes, last question on the slide.
Regarding the performance of the transcriptional regulation versus macroRNA. I mean, the TF versus macroRNA, right? I looked at the TF explains very well across different tissues compared to the macroRNA. I mean, macroRNA regulation looks very tissue specific. Is there any biological reason for that? For me, it kind of looks counterintuitive because I assume that transcriptional regulation should be way more tissue specific than post-transcriptional regulation. What do you think of that?
So this, I think there's a slight nuance to your question that this doesn't actually say that there are that there's the same microRNAs and they should be affecting the same gene. So this could be different sets of microRNAs because we are doing independent models per tissue. So those models are not directly comparable. And there will, of course, be variation across the regulation across tissues. And we know that we've seen it from much simpler settings when you just look at microRNAs. So there is I feel like this is one of the benefits of doing this kind of work or doing any other integrative type of work is that we see all the different factors, well, the 6 different types of factors and how they work together to make a certain decision and what actually gives us additional explanatory power and what doesn't. And maybe what you said is one of these beliefs that we get from looking at each of the individual type of regulator independently, but this allows us to draw more complex conclusions. So this is good, thank you. So one of the things, actually, to your point, is that EGFR gene looks like it has a really complex pattern of regulation. So what this means is that you have this variation across tissues, and variation in some tissues, there seems to be a dominant effect by transcription factors. In other tissues, there seems to be a dominant effect by microRNAs, but it is quite clear that it is this multiple contributors that are actually making a difference in regulation of this specific gene, which kind of makes sense for it. The lefty one, my favoritely named gene, is actually has a much more quiet type of regulation. You can see that the patterns, at least here, from this data appear to be much simpler. So in sarcoma, it looks like microRNAs are very, very important. Link RNAs are important across multiple tissues, and the CNVs I've already mentioned. So there is this combination of the different factors that gives you a sense of a much fuller sense of the regulation of the gene. And of course, we have this information on more than 10,000 genes. It's over 16,000 genes if you look at kind of non-overlapping genes across tissues. So how could you use it if you don't really care about how genes are regulated? Of course, everybody should care about how genes are regulated. But suppose you are doing EWAS study, and you have identified some of the CPG sites that seem to be important for your phenotype. So these are your top significant hits, and you are trying to figure out which genes they map to. This, in fact, is the exercise we did. We took Nature Communications paper that published a hit of EWAS probes, and you notice that the very top, actually I have this thing. So the very top hit doesn't have a corresponding gene. So they couldn't map it to a particular gene. And it makes sense because if you look, this gene is actually in a very kind of high density area on one of those smaller chromosomes where it's really hard to annotate. There is a closest gene, but there are tons of other genes. So what we can do is we can actually look at where this gene, this probe has contributed the most, to which gene expression it has contributed the most. And look at the top, in this case, 5 genes, the top 5 genes, where this gene had the most contribution across tissues. And it looks like the zinc finger 625 was that gene. The way that we do it is very simple. We take the coefficient of the particular probe, we weigh it by the quality of the model, how predictive the model was, and we sum it up across all models.
Should we be worried about the y-axis?
About how tiny the scores are. You can if you want to, but it's the goal of this exercise was just ranking. So there is some evidence. This is a score, right, which is weighed by the coefficients in the model. There are many microRNAs in the model, so these coefficients are going to be small. So this is not 0 to 1 kind of thing. This is not scaled.
What are typical scores? Is this typically high, or is this?
That's a good question. I don't know. We really treated it as a ranking exercise, but. Yes, if one wanted to actually report this as the closest gene, then we will, Lauren, I see you taking a note, to actually make sure that we report the ranges and the important ranges across, and the average across all genes.
Typical for however many regulators, so that we know if it's higher than normal.
Yeah. To report the summary stats for this statistic, yeah. Yes?
How do you think measurement uncertainty changes the results, or how does that play into the size of error bars that you would put on things? You know, like
Difficult question. Measurement uncertainty. It obviously plays a lot. If we can trust the measurement and we still use it, right? I mean, we don't know not to trust it, and we still use it. That's why we had a really rigorous QC. But yes, I, as a computer scientist, I'm always suspicious of biological measurements. So it is difficult. And the uncertainty of the measurement can't even be quantified. Why did you ask such a difficult question? So it can't even be quantified, right? So we can incorporate the uncertainty on the coefficient estimates from the model. So we can do that. But this is just a relative error. So it is a very important question that I don't think we can address with this data, sadly. All right. So just to finish up that vignette, this is where the probe is. This is the zinc finger 625. The other genes are not in such close proximity, but they were also nearby, but they weren't ranked as highly. So what we could also do is we could take the full list, and just like people do functional annotation, we could actually do implication of this list on the top gene, on the ranking of the genes. So here we take the full list and we see how these probes contribute to the gene expression variation across tissues, across genes. So this is done by computing this gene score, which is essentially the information about how important this probe is for the phenotype that we care about, and weighed by the regulation score that I just described. So the gene score will be a sum across cancers, across tissues. Yeah.
I have a question. It's a little related to my previous one, which is, how often will your model basically say, oh, actually, I don't know? Like, basically, we don't have enough information in the data to say something, because, again, of course, we haven't yet measured everything with biology. Yes. And it would be nice to not just say, oh, here it is, based on the very limited data that we have, but to sometimes say, the data is actually not sufficiently predictive, so I'm not going to say anything.
So you are absolutely right. This is the answer to the previous question would help to fix the answer to this question. but there is so for me, personally, before we even do that, if there were a uniform distribution across many genes, then that would, to me, would mean that we don't have enough evidence for the contribution. So it's just the simplest analysis. I don't like it as much as the kind of the analysis of trying to figure out what are the implications to the random genes and looking at the empirical distribution of that and trying to figure out where this, how likely this hit is actually to be associated with the gene.
Model and basically saying, you know, this is so much more likely, I think would be one way to do that.
Yeah. This is being done. This, you see? It's being done.
The act of writing on the paper is
well, but we can do everything that we can think of. So this is great. Yes, thank you. So this is, I mean, the usual storytelling, which I might go quickly over. So the genes, the original study had to do with a kidney function, and the genes that were in the top 3 were already implicated in renal or renal fibrosis or renal function, which is comforting, but of course it's just a story. So it would need to be followed up by a functional experiment.
I'm so excited, that's why I keep asking questions. I would love to know if you were to rank genes based on their predictability or predictiveness, and if you were to rank genes based on the number of citations, and you show a scatter plot of that, it could tell us all, go study that gene, seems to be very important, but totally understudied. Have you tried that?
So I think this is tricky because importance of a gene with respect to what, right? There are many phenotypes out there. There are many for which some of the genes are important. We know that there are a lot of understudied genes. We can see them. But figuring out the importance of the gene and implication of the gene, I don't know, it's going to be hard to do because we don't have the range of the phenotypes that we have associated with this data. And you see my point. So this is difficult. I think we are hoping that the way that it will work is that now people who are discovering genes that they don't know anything about, they will not go to their favorite gene, but they will actually look at it using this resource and then decide. I very, very frequently, I am at the poster and I see a list of genes and the biologists present and they point to the middle and they say, this gene, it was this gene. How do you know? So for me, hopefully this helps with kind of the standardization of that analysis for me, because I don't have the background that the biologists have. So to summarize, we have 20 minutes. I have 2 more topics to cover. But to summarize, what we have done is that this is an immodest resource that we have developed. And we can help to annotate hits. We can help to, with a kind of experimental design and planning if somebody wants to study gene regulation. And what is also exciting is even though we built these models in cancer data, we actually use the cancer data to predict expression in healthy tissue or nearby tissue. And 43% of the genes had 70% of explainability of their gene expression in the non-cancer tissue, which I think is very nice. So we don't expect them to be. Of course, a lot of it is cancer dependent, but it's nice to see that some of these models actually translate. And then in healthy tissues, the EQTLs come up a lot more. So these are the same models, but the EQTLs are much more predictive of expression. So this part of my talk is a very important message that I have to give, and I'm going to use a Lee-Fraumeni syndrome example to do this. So for those of you who don't know, Lee-Fraumeni is a hereditary cancer predisposition syndrome. So people with germline TP53 are likely to get cancer throughout their lifetime. P53 tumor suppressor is associated with 80% of clinically diagnosed cases. And it's really dramatic. This phenotype of having that mutation is really, well, actually, range of mutations in TP53 is really dramatic. It's the 40% chance of getting cancer before the age of 25, almost 100% over lifetime. So these families have really brutal stories and histories, and I work with a clinician who works with these families and is collecting this data. The only hope to actually help this, and they get multiple, so there's a primary, secondary, tertiary tumors. They get multiple tumors over time. They start getting them early. And the question is, the earliest we detect, or then the better it is for the outcome, really. So this is our database. Quite sadly, you can see that a lot of them are children. A lot of the people in our database are children that get cancer. So this is the age of getting the primary tumor. And you get cancers. So these kids, they get cancers almost anywhere. So it's very hard to track. ACC is the most predominant in our database, but there are many. And
sorry,
yeah.
You are in a kids hospital. Is this why your distribution is skewed? Because I would have expected 40% by 25, and I see 60% by 25.
It is a little bit skewed to that, but it seems that anecdotally that the phenotype becomes harder as more generations. So if a mother has breast cancer at the age of 28, she might have a child that will have ACC at the age of 8, and then a younger child that will have a cancer at the age of 5. So it's a very unfortunate phenotype. So there is a surveillance protocol. It's called Toronto Surveillance Protocol because now it's used around the world. That was created by Dr. Malkin, who is my very close collaborator. And they do measurements, they do ultrasounds, whole body MRI, blood work every 4 months, just to try to detect it early. There are benefits already, so they show really greatly increased survival and early detection of cancer. So this really, really works. But there are challenges, and the challenges mainly come from a burden to the families. So every 4 months, a parent has to bring their kid to see if they have cancer. You can imagine, it's psychologically very difficult. And also, it's very hard to image small kids. So if they suspect that there is cancer, you have to do this whole body MRI, and it's just hard to do on 2- and 3-year-old kids. For those of you who have kids will know exactly what I mean. So they move a lot. So David has proposed this clinical pipeline. So we get methylation data with draw blood, get methylation data, and kind of build a predictive algorithm to see whether the kid is likely to get cancer before the age of 6 from methylation data. We built such a model. This is a computational pipeline. So first we did pretty rigorous feature selection because we have relatively small sample size. So we first found all the regions that are specific to the LFS patients, so TP53 carriers versus not. Then, of those regions, we removed all the cancers. So these are the people with a TP53 mutation both, but some of them have cancer already and some of them don't. So we had to remove all of those regions because we want predisposition. And then we just built a classification algorithm, which in this case was random forest, and to basically predict from the data whether this methylation regions in the methylation profiles in these patients are likely to have cancer before the age of 6. It turned out that the method performs fairly well. I was more worried about it until I saw other work that takes methylation data and actually predicts cancer onset 5 years ahead. I don't know if you know of that work. There's a company in San Diego that I saw at the SHG a couple of years ago that had similar results. So that made me think that methylation does carry some of the signal. In this particular case, this is believed due to the fact that TP53 might alter the epigenetic landscape, the methylation landscape of the genome-wide. So we have the 78% sensitivity, about 87% specificity, and on the null set, on the individuals that we had methylation data that didn't have cancer for either before or after, it seemed to perform well. This is a result on a very small data set, so with a caveat, we have to do prospective trials and everything, but we are hopeful, basically. And then, of course, the clinicians are saying, okay, what are the probes that are causing this? And when we clustered the data, so this is the important point, actually. So when we clustered the data, we saw these clusters. So these are patients, and this is methylation. There are 1, 2, 3, 4, 5 clusters in the methylation data. So what does this look like to you? This is genome-wide signal. We have areas and regions in our methylation probes that were selected that seem to be very highly correlated.
I'm not sure I understand. Are these the probes that are predictive, or are they just all the probes?
This is all the probes that the model did not select in the top whatever category, right? This is what. So no, this is before the selection. So this is all the probes that are in the regions that we cared about that were put in the model.
But then you're also going to show us the ones that the model selected.
So what we did was they asked us to show the probes that were selected. And we did this instead. So we took the sampled probes representing each of the 5 methylation regions at random 100 times. So what happened was when you had a cluster representation in your data, you selected the same amount of probe as in the winning model, we had statistically identical results. So, identical, statistically non-significantly different results. When there was no representation of the clusters by the probes. So if you selected the same number of probes but didn't respect this kind of the genome-wide pattern in the methylation data, we had worse results. So basically, it's this pattern that is being captured. And the point here was that they were looking for the probes to design this kind of assay. And I'm really worried that because our data was very, very small, we would have honed in on some probes that are not really generalizable to the full population. And yeah.
Are the 5 classes that you show corresponding to different chromatin states or something?
We didn't go that far. This was a clinical kind of paper.
Promoter will be always off, enhancer will be somewhere in the middle, etc.
It could be. They are broad enough that it's probably a more broad pattern even than that. That would be my guess. But the point is that I see very frequently that people take a small data and look at a model that has regularization and the top genes, and don't necessarily look at, others. Did the model actually have a chance to identify the genes, or is this, a numerical exploration of a multimodal state? And I think in this case, the result was that we believe that there is a general pattern in a methylation spectrum, and that's what gives us the predictive ability to predict the clinical outcome. And so, in my case, I would recommend such analysis, and actually had a talk by Rob Tibshirani recently, and he also was recommending this type of analysis, so which made me feel good. But
now, I mean, what I was hoping to see is some kind of case control, and here are the probes that are very different or something.
I don't think this is the case. We don't have enough data to make that statement, and I think this is the experiment that we try to do, to say these probes, there are enough sets of probes that we cannot distinguish between, given our data. So this is a critical case. You can design an assay in a different kind of way, serving this clusters. Maybe it's the chromatin. So more annotation of that. But right now, at the probe level, we cannot point to probes that would implicate the regions. So I'm going to be mindful of time. We have about 10 minutes. Yes? So I will go fast. I might not take as many questions. So please ask clarification questions only for now. And then I think we have the discussion time after the talk. And I will be happy to answer whichever questions you have. Yeah? So This is a drug response prediction problem that we've been working for several years on. And I want to share these results with you because this is a space where predictive models are very commonly used. It's a problem that is explored well in the literature, and I think our results are interesting. So quickly, the samples that are coming out of clinical trials that have biological data, such as gene expression, to predict drug response are very small. So it's very difficult to build kind of complex models that might be there that map from gene expression to the phenotype, the drug response, to be able to build a good predictor. So the idea is that we have a lot more data on preclinical models, such as cell lines or PDX, not as much on PDX, but cell lines definitely, much of it collected here, that we could use to make that prediction happen, and then do the domain adaptation that Lauren so eloquently talked about. So quickly, as a computer scientist, I like to see every problem represented as a matrix and outcome. So here we just took 1,000 landmark genes. We didn't have to take 1,000 landmark genes, but this is there is a reason for that. It's about 600 cell lines per drug. We selected the 26 FDA-approved drugs, and we were mapping to response, non-response. So again, a caveat, the outcome in itself is tricky. You have to threshold this kind of curves that you get out of the lab and say this is response, this is non-response. There are standard ways to doing that. All of them are, have their caveats. So with those caveats in mind, we did have a response, non-response vector, and went on to look at how to make this prediction. So these are the results from the classical models for this data. So random forest seems to be performing the best, almost at 0.7, not quite. The data that we used was the CTRPV2 database coming from here. If you take the 100 top PCs in the data, you definitely help the ridge regression to do a little bit better than the random forest using the raw data. So it seems that there is enough of noise in the data that perhaps a dimensionality reduction first might be preferable. So this is our current kind of best straw man on making the prediction. And then my student said, but I want to use deep learning. And I said, OK, let's use deep learning. It's a small data set. This is going to be difficult, but here are all the things that he's done. Yes, it took years. So hopeless overfitting, but by majority of the networks. Feed-forward networks overfit. Mini batches don't help the regularization. Siamese networks, where you have two networks and you're just trying to see make a similar prediction for one as if you have for another. Overfit. Adversarial domain adaptation basically was a really difficult to balance out all the regularization criteria, and it seemed that we removed all the signal when we did proper regularization. So fortunately, there's this really nice, though a little bit finicky, variational autoencoders. How many are familiar with variational autoencoders? OK, great. So no big introduction, except that I love them because they're graphical models with nonlinear functions. This is graphical model. The paradigm of graphical models is huge. It's very, very powerful. You can represent all kinds of basically relations between variables and encode them. Here, this is rather simple. We have gene expression, which is our X. We have latent representation of this gene expression. And we have the outcome, the binary Y, which is shaded, half shaded because some of them are unobserved, so semi-supervised. And this is the latent representation of all the kind of variation that is not explained and not captured by the drug response. So this is each of these kind of edges is a neural net, which we fit to our data. This is the SSV, the semi-supervised variational autoencoder that was proposed in Enigma. It does a little bit better than the ridge regression with the PCAs. But we remember that we have more data. And as you notice, a big part of this talk is kind of trying to bring more data together to make informed decisions and build better models. So here, there's also this links connectivity map database, which a lot of people know is quite noisy, but they do have gene expression before and after drug treatment. So this is satisfying in a way because there are fewer arbitrary decisions made and thresholding the response, non-response, right? It's in the regional domain. We had gene expression before, we tested the drug, we applied the drug, we have the gene expression after. I think it's more satisfying because the domain is the same. So we can build variational autoencoder here. So this is before treatment, this is after treatment. Again, this database is relatively limited, so semi-supervised. And this link from Z1 to Z2, there's a latent representation before treatment, latent representation after treatment. And this is the function that encodes the drug effect. Okay, this is what we are trying to learn. We tried multiple functions here. We tried neural nets here. Did not have enough data to fit it. So we tried identity function, and it seemed like that performed worse than a linear function. So this is a linear function right here. So this is what we did. This is our data encoder for the pre-treatment, decoder for the post-treatment, and this is our drug response VAE, Dr. VAE, which is the full model that takes advantage of both the pre-treatment expression, post-treatment expression, and tries to predict response. So this model performed a little bit better, but note that the scale is really granular. So yes, it is a little bit better, and it is significant. We did test significance by resampling, but it's not an amazing improvement. But what makes me really excited is that this is not a very large data set. So generally, I would stay away from deep learning for this data set, but because this variational autoencoder is essentially kind of a graphical model, and we can play with the architecture and a regularization, we can start building such models. What it allows us is the flexibility of bringing all these multiple resources, which I really like. Yeah?
So you just still train on the hundreds of PCs?
No. Raw data.
Raw means 978 genes, or the 12,000?
Yeah, 978 genes. The reason for that, I forgot to mention, is because the links is only done on 1,000 genes, on the L1000. So that was the intersection that we had.
Level 2 or level 5? What is the level 2? Do you remember which one?
No.
Okay.
Sorry. So quickly, this was the result. I do have a domain adaptation of trying to move this result to the patient data. This would take another 5 to 10 minutes, but we are at 11 o'clock, so I'm happy to stop. I'm also happy to mention some of the other projects that we do just by name so that you have an idea. But I want feedback. This is 11.
No, I think one more minute, and then we're gonna have to let people go out who need to leave at 11, and then we can definitely take a little time to go over all that you didn't get to that you wanted during the discussion.
All right. So I will just give you a takeaway from the domain adaptation that the way that the cell lines work and the way that the response in cell lines is captured, the way the response in patients is captured, and the lack of, I'll just point out to this. These are patients and these are cell lines. The fact that they really don't jam, talking about the overlap and support in Lauren's talk, makes me think that the domain adaptation in cell lines, as we imagine, kind of just taking models built on cell lines and domain adapting them to patients, does not work. And we have reasons, the theoretical reasons that were discussed before that allow that. Basically, adding patients to the trained cell line model does a little bit better, but when you just train models on patients to patients, it does better. So there is no point of adding cell lines or any kind of transference. So this is my conclusion. Just to give you a sense of some of the other projects that we have, We are working on predicting of cardiac arrest. We got a grant to implement it in a hospital, which I'm super excited about because there's so much more to machine learning and development. We published on image saliency, so a new counterfactual approach to imputing on images, but we have a reinforcement learning paper for figuring out which measurements to collect when in critical care. We are working a lot on explainability, so I believe right now is the deadline fly clear, so at 11 o'clock, submitted the last version. It's on explainability in time series using counterfactual models. I'm sure it will not be accepted, but I still think it's a good paper. So this is a lot of the work that we are doing is around building models that will actually be adopted. So we want to make sure that what we are optimizing is something that the end user will actually care about. So we actually have done surveys of clinicians about what they want to see in a model and what they want to optimize, which I think is very exciting. There's the surgery in prenatal hydronephrosis. Lauren is flying today to Arizona to give a talk at the urology meeting on this. This is predicting directly from ultrasounds whether or not to do a surgery for prenatal hydronephrosis. And just on Sunday, we got this funded. It's very exciting. So this is also for the Lee-Fraumeni syndrome. This is building GAN models and augmenting the whole body MRI spectrum, because in kids, it's very small data sets. There's no way we could ever do any kind of complex imaging techniques using just existing data. So we are building out the resources for dedicated MRIs. We're building resources for whole body MRIs, so generating whole body MRIs, to try to improve the cancer surveillance part of the existing protocol in the hospital. So with that, I thank you for listening. I'm sorry for running late. And my amazing, amazing lab, of which there are a few people, it's a little bit bigger, and also my funders.